and breast cancer in the liver. The worst types of cancer are the metastasis ones or the malignant ones because once it's invaded the entire body, that's when it's harder to get rid of. Not impossible, it's just harder to get rid of. And the benign types of tumors are the easier ones to get rid of. And we'll learn later that the best type of survival rate for any cancer is earlier detection. That way, if you find it early enough and it's small enough, you can very easily remove it. So we do have other types of cancers. Technically, some of the benign cancers in the body, they're not a worry or a concern. So most moles or warts or skin tags, those growths on your skin, they're technically benign cancers because they're a little bit out of control, but not completely where they're metastatic. Cysts are also another type of cancerous growth. So what a cyst is, it's a big ball, and this ball fills up with fluid. People get their cysts drained over and over again, and if they want the cyst removed, they have to get every part of the cyst out of their body because even if one fragment's left, the cyst will grow right back. So it's very similar to a wart or a mole. In order to get rid of a wart or a mole, you have to get rid of that cell that's lost mitosis or control mitosis. That way, it won't grow back. So most people just take the top layer of the mole off. It'll grow right back. Okay, so with risk factors for cancer, a risk factor is just anything that will cause a disease. It just increases the likelihood of that disease. So in this case, the disease that we're talking about is going to be cancer. There's two main types. We have the inherited type, which is going to be found in genes. So this is based on somebody's DNA. And we have the environmental type, which is going to be based on factors like chemicals or radiation or something in the environment that's being put into the body to cause cancer. So with the inherited or the DNA based gene types of cancers, just because somebody has a gene that's correlated with cancer does not mean that they will get cancer. So I think I talked about this before, but there's a famous one. It's called BRCA1. So BRCA1 stands for the breast cancer 1 gene. We also have a BRCA2 and a BRCA3. But with BRCA1, it's a gene that when it's working correctly, it's going to stop cancer. But the BRCA1 gene is very close to mutating to a form where it doesn't work anymore. And if somebody has that form, it doesn't mean that it will mutate to cause cancer. It's a higher chance. So again, I talked about this before, but with Angelina Jolie, the actress, she screened herself. She supplied her DNA. They looked at her DNA. They found out she had the BRCA1 gene. And that information, plus the fact that her mother and her grandmother both died of two different cancers, breast cancer and ovarian cancer, she decided to actually have her breast tissue and her ovaries removed because that's what the BRCA gene affects the most. So just because she has that gene, it doesn't mean that she would have gotten cancer, but she took that precaution anyway to protect herself just in case. So again, that's an option out there. Just that's want to make it known that if somebody has the gene, does not mean they will get cancer though. And then we have the environmental exposures. So these are from chemicals or substances in nature. There's special types of substances called carcinogens. So a carcinogen, whenever you see that CAR, so CAR, that references cancer. So this is a cancer-causing material. It's a special type of mutagen, and a mutagen just mutates DNA. So with these carcinogens, they range from all sorts of things with chemicals, some types of food. Let's see, sunlight is a good example too. So what they're doing is they're affecting the rate of mutation in DNA. They have some effect on DNA that's either going to rearrange the nucleotides, break it up, and then from there, the DNA is going to mutate. So on their own, carcinogens have a low effect. But if you start combining carcinogens, it's got a synergistic effect. So with synergistic effects, two or more are going to add to the increase. So examples, we have alcohol, cigarette smoke. So both of these by themselves are carcinogens. So if I have person A who's a smoker and person B who drinks alcohol a lot, They both have their own chances of getting cancer just because both of those molecules are carcinogens. Now, if I have person C, who's a smoker and a drinker, person C has an even higher chance than A or B just because of the synergistic effect of these alcohol and the cigarette smoke working together to increase the rate of mutation. Uh, Looking at the human population in general, the most common type of cancer within the human population is going to be skin cancer. And that's because every human is exposed to roughly the same amount of carcinogen material when it comes to sunlight. So somebody's outside during the day, they're getting sunlight hitting their skin, and the sunlight with that electromagnetic radiation, it's enough energy that it goes down into your cells, and it's going to go into the nucleus of the cell. And then you have your DNA, right? Remember, your DNA is that double-stranded helix. So this strand is being held together by hydrogen bonds. And we talked about denaturation of a protein, right? So you just put a little bit of heat in there and the protein unravels. Same thing with DNA. If I put a little bit of heat in my DNA, it's actually going to unravel and break apart. So we can very quickly put it back together, but not every time do we put it back together correctly. So if we put it back together incorrectly, that's when the cells mutate and the skin cell will become cancerous.
Now, in terms of the cigarette, okay, it is not necessarily the one that smoke it, or if we close to those, smokers, we also can get that effect.
You are correct. Yeah. So it's not just not just the smoker, it's the secondhand cigarette smoke too. So if somebody's around you smoking, that's just as bad as smoking the cigarette.
Okay.
Yes.
So that carry out from the breathe, the air that we breathe, right? So that affect, okay, through the lung, and then it become toxic for some part.
You are correct. Yes.
Yeah.
So like it was about 20 years ago, it was perfectly fine to smoke in public here in the US, right? And then we figure out secondhand smoke is a carcinogen and does cause cancer. So a lot of public areas just don't allow smoking at all anymore. They have their own little designated smoking area way away from the public, just because it's such a high risk factor.
Yeah. So this basically affect the lung or lung cancer.
Lung cancer, throat cancer, nasal cancer. Yeah, anywhere that smoke's touching.
Oh, so
Yeah,
it is
not
not just
yeah,
lung, but it affect the whole thing.
correct. My aunt, she died of lung cancer. I mean, not lung, throat cancer because she was a smoker.
Mm.
Mm-hmm.
Now, this is very important in developing countries, especially in Indonesia. Okay, it's smoking like drinking Coke. Okay, it's scary. All right,
Uh-huh.
so this kid need to be able to tell everybody over there. Okay, but in the U.S., all right, the law, the rule is very clear. If you smoke, go outside the
Yes.
building, but not there.
And there's 100% no smoking at a hospital. No.
Right.
Do they
But
allow do they allow smoking in doctors' offices and hospitals over in Indonesia?
I'm not sure that. Let's ask the Alfonso. What do you think? I think maybe in the hospital there may be more restriction, but outside that hospital, people are smoking everywhere. I'm telling you, that is the biggest industry in Indonesia.
Yes, it makes sense. It's not only is it you can make money off of it, it's very addicting. So people get addicted to nicotine and they crave it. So that yeah.
Yeah, yeah. So Indah and Hani and also Alfonso. Okay, if you Alfonso, if you put your cam, how three of you guys need to start telling your family members how that smoking will affect not only the smoker but the whole family. They even smoke in eh, the house, okay, with the AC on, okay, air conditioning on. Okay, it means what? There's no outside air. It's only the air, filled with smoke and poison. They don't know that. I mean, it's just totally insane. Okay. So
So
this is,
the reason why we know secondhand smoke is a carcinogen, it's a very easy test. So they take some bacteria and they have it on a petri dish, and then they actually have to feed the bacteria. So if they limit a special type of food to the bacteria, it dies. So they take that
Mhm,
same bacteria and put it on another petri dish. They
mhm,
limit the food, but they put in the secondhand, like in this case, the secondhand smoke, and they survive. That means the DNA mutated, and that's what it's
mhm.
doing. It's actually mutating the DNA inside the cells, causing it to change the DNA sequence.
Yeah.
That's why it's so dangerous to our cells, because when it mutates, it makes the cell cancerous.
Yeah, that's very scary. The smoke can change the DNA structure of a
Mm-hmm.
person. I mean, it's just this is more like a public safety issue, okay? That none of them in Indonesia or most developing country really understood this kind of thing, okay? I mean, it's just a very simple thing, okay? But yet, people do not understand this. How can we tell people, hey, quit smoking?
It's still, it's still a problem here in the US too, because smoking is still a major profit for the US.
Yeah,
And a lot of people, cigarettes are like 20 bucks a carton, if not more. So people are
yeah,
still going to that, paying that much money, and they know that it's a carcinogen, but they still can't stop themselves.
right, yeah, yeah. Maybe we need to write an article, okay? It less academic, but you know, write an article where ordinary people can read it, understand it, and put with the data. Okay, because they not even know what DNA is. I
Mhm.
did not know until I follow your class right here. Okay, then I say, oh my goodness, this is a big deal.
It is, yes. So this is when I have my students, a lot of them in uh, the satellite campuses like Lawrenceburg and Lewisburg.
Mhm,
There's a lot of
mhm.
smokers there for some reason,
Oh
and
my,
you can smell them. You can smell the smoke on them.
yeah,
And
yeah,
then
yeah.
going into this, it kind of is an eye opener for them.
So have you discussed this thing to them or with them?
Yes, we talked about this week actually. So
Uh-huh. So is there any behavior change?
I've had a couple. Yeah, I've had a couple stop. Vaping is also pretty bad. If you know anything about vaping, it's
Mm,
not lung cancerous, but it does have its own body physiological uh
yeah, yeah,
downsides.
yeah. So DNA can also bring a fact for women, okay, the ability for her productive, right?
Mm-hmm.
My. Hmm. Wow. Thank
Okay,
you, Ken.
you're welcome. So let's talk about mitosis. We're going to focus on this one today, mitosis. So that's a type of asexual reproduction, meaning that when the cell goes and makes a copy, it's an identical copy. There's another type we talked about, or we will talk about, meiosis. So they sound very similar, but with meiosis, it's a form of sexual reproduction. So the individual is going to make a sperm and an egg cell, and then when those two cells come together, you got a completely new individual. So they're not identical copies. It's This is easy to do with mitosis. It's pretty easy, but with meiosis, it's important for variants. You're going to change up things and make new genes, make new individuals. That's why meiosis is important. But with mitosis, it's important for growing, getting bigger, for repairing, healing, stuff like that.
Mhm.
Uh, let's see. We need to talk about chromosomes and genes before we look at the mitosis stages. So with DNA, remember DNA is held inside the nucleus, and then most of the time DNA is in a form called chromatin. So chromatin is the loose, uncondensed form of DNA. So if I was to draw out the long double-stranded helix, it would be just this long linear structure, non-condensed. So that's chromatin. The problem with chromatin is if you want to move it, which you have to move it during mitosis, if you pull on it and there's more chromatin around it, it'd be like pulling long threads and they're going to get tangled up with each other. And if they're tangled up and they make a knot and you pull on that knot and you break the DNA, that's a bad thing. We don't want to be breaking or tearing our DNA. So in order to move the DNA during mitosis, we're going to take that chromatin and we're going to densely compact it into a long linear structure like this. And that's a chromosome. So with this image in the bottom right, the colors in the sphere, that's chromatin inside the nucleus. And they've been stained different colors to show you where the chromatins, the specific strands are for that DNA. Now, these over here are the chromosomes. So the chromatin condensed down into these chromosomes, and the colors are matching where they go. So each one of those colors, they're going to condense down into one of these long chromosomes. And then you should only see these chromosomes during mitosis. Every other time in the cell cycle or the cell's lifespan, it's going to be the chromatin or the loose form. Now, since we're going to split the cell and make two cells during mitosis, you need to copy the DNA. Let's say that we didn't copy the DNA first. So if we were half the original amount, we would have half the amount in both cells. And then from there, they're going to go through mitosis again. So if we divided without doubling 4 cells, we have a quarter of the DNA in each cell. And if you keep doing this over time, you're eventually going to run out of DNA because you can't half forever. But if you double right before you half, then you will always have the same amount of DNA. So right before you go through mitosis, you go through this phase where you double the DNA, and then you have a chromosome, and then you have a copy of the chromosome. So they look like this. And both of these structures are called sister chromatids. So these two are copies of each other. Right in the middle, we get a little dot there going. Right here in the middle, they're attached with what's called a centromere. The centromere is the area where they attach, and then coming off of the centromere, you have these little hooks. And these hooks are called kinetochores. The kinetochores are going to be important for when we move the chromosome around. That's the site of attachment. Think of that like a truck hitch or a train hitch. They're going to attach to the hitch, and then they can push and pull the chromosome around.
Mhm. So what does it mean
What does what
when they
mean?
attach it together?
Why they're attached together?
Yeah, when they join, right? The two chromosomes are joined by centromere.
Yes.
Okay. What does it mean when it joined?
It just means that they're copies of each other. Um, they're so every organism has a different number of chromosomes. Like this
Mm-hmm.
individual right here, each one of these individual colors is a different chromosome.
Mm-hmm.
And then right now it's not copied. So these chromosomes don't have any sisters. But if they
Mm.
did have a sister, there would be another one attached to it like that.
Okay. So where did sister coming from?
Right here, right here. So before you go through mitosis, you go through this phase of duplication. So again,
Mm-hmm.
we have our long strand of DNA, right? It's in this form,
Mm.
a
Okay.
double-stranded helix.
Yeah.
So you can't copy it in that helix structure. You actually have to straighten it out like this over here. So you have to straighten it out, get rid of that spiral, and then once it's straightened out, you will unzip it. So this enzyme that comes in is called helicase. Helicase is going to go into the middle of my DNA, and then it's going to unzip my 2 strands. And it kind of opens them up and separates them.
Mm-hmm.
Now, the blues that you see, these blue structures,
Yeah.
they're going to be my parent strands, also sometimes referred to as my template strands, because that's what we're using to make a copy. And then right here, these 2 little rectangles are an enzyme called DNA polymerase. So DNA polymerase's job, it reads the parent strand, and as it reads the parent strand, it looks at the nucleotides, and it's going to put my complements to the nucleotide. So let's say that I start here. Actually, you know what? Let me grab a blank slide. Need some more room.
Uh-huh.
So just a reminder, when we're looking at DNA, we're going to have nucleotides that are adenine and thymine, and then guanine and cytosine. So these are our complements. So if I'm looking at my bottom strand first, T, C, G, the strand's going to be separated, and then I'm going to have DNA polymerase come in right here, and it's going to start going in this direction. And it's going to look at the bottom strand. So the first thing it comes to is this A. So as
Mm-hmm.
it hits that A, it's going to, hey, what needs to be my nucleotide on the new strand? So the complement of A is up here. It's going to be a T. When it gets to this T right here, it goes, what's my complement? It should be A. When it hits this C right here, it's going to be a G. And then when it hits this G right here, it's going to be a C. So this DNA polymerase reads the bottom strand. And then up here at the top, another one will come in. Well, let's see. Let's do T A G C.
See?
And then it'll start right about here. It goes in this direction. And it's going to read these nucleotides in that direction. So the first one it comes to is this C. So the complement there will be G. It's that G, the complement will be C. It hits that A, the complement will be T. And then it hits that T, my complement's going to be A. And if you notice, the strands are now identical. We have TA on the right, this right area right here. Down here we have TA. And then we have AT, AT, GC, GC, CG, CG. So when we're through with this whole process, we're going to have two strands of DNA that are identical copies of each other. And then when we create them, that's when they join together at that centromere, that middle area in the middle.
What's the big deal about this joining?
Okay, when there's a phase of mitosis called anaphase. With anaphase, you want one sister to go to opposite sides of the cell. So
Mm-hmm.
if I attach them together, and then I have a microtubule from both sides of the cell join, only one's going to be able to join to both sisters. If they
Mm-hmm.
work together, that means that there's a likelihood that they're both going to be pulled to the same area.
Mm-hmm. Mm-hmm.
We don't want that. We want one DNA sequence to go to one end of the cell, and we want the other DNA sequence to go to the other end of the cell.
Mm-hmm. Mm-hmm. Okay.
Now, there's a good question for next week too, because with meiosis is randomized, so that might actually have a little play on it next week when we talk about
Very
meiosis.
difficult. Yeah,
Yes.
very difficult to predict. Mm.
So with mitosis, it is a part of a larger cell cycle. The cell cycle has two very big phases. You have interphase and mitosis. So if you look at the big circle on the edges, you see interphase, 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 and then you see mitosis. So those are my two main phases. And you notice that interphase is three of those. So interphase should be the majority of the time spent with a cell. And then mitosis is a very small little gap here, which means mitosis is very quick compared to the life cycle of a cell.
Mm-hmm.
Your cells that are doing mitosis are special types of cells called somatic cells. And a somatic cell is going to be every cell in the body except for cells that are involved in sex cell reproduction. So most of the cells in your body are somatic and should have went through mitosis or are still going through mitosis. So we'll focus on interphase for 1 second before we look at what's happening during mitosis. So if you look at interphase, you have a G1, an S phase, and a G2 phase. So G1 is standing, or it means growth 1, gap 1. And during this time, the cell's job is to grow. So if right here at the end of this cycle was a process called cytokinesis. So cytokinesis means the cell half the original size. You had a nice huge cell, and then during cytokinesis, it's cut in half. So you have two small cells now. So the whole purpose of G1 is to get back to that original size. It's going to grow. It's going to make proteins. It's going to make more plasma membrane components, and it's going to start to get bigger. Now, cells don't like to get too big. So once it hits a certain size, it's going to go, hey, I need to go through mitosis again because I don't need to get any bigger. So that's what G1 does. So when it hits that certain size and it wants to go through mitosis, it's got to go through the S phase. So this is where you double the DNA. So what I just went over, that's what it would do. It would make 2 copies of every chromosome. So it had a sister for each pair. And then after the synthesis phase, it's going to get ready to go through mitosis. Before it does that, it goes to the GAP2 phase. So GAP2 says prepares for mitosis. What it does, it harvests materials for microtubules. It's going to move around some organelles, try to kind of equal out the cytoplasm. So that way it's going to be equal when it splits. It stores things it needs in order to prepare for chromosomes. And then once it has everything it needs, that's when it's finally going to go into this mitotic stage here at the end of GAP2.
Now, this is the question. All right. Let's relate it with the problem is some people are hyperactive, okay, like ADHD, okay. Now, during this process, okay, which one of the 3 from this cell growth, okay, and DNA that will cause this some of the kid become super active? Why
Um,
is that?
I don't I don't think that would have any effect with mitosis. ADHD, hyper attention deficit disorder, it's a behavioral issue. So it might be more related to brain chemistry
Oh,
and
brain.
neuron. Yeah.
Now, even brain, okay, if you look at this, during this process, even brain, okay, was growth, right? Or form, isn't
Mhm.
it?
Yeah, your brain grows.
Right? So
Mhm.
therefore, you know, what we call it disequilibrium. Anything is not in equilibrium or, in equal sign will make, the left may be heavier than the right. Okay, it is not equal. Okay, now this is the subject of, it's an incredible opportunity for anyone, okay, to do this research, especially brain related like ADHD and some other thing, okay, and not, and the other problem like single cell, all right, the blood cell, okay, for some people, especially for the, Black American, I think the single cell problem is more, common, okay,
Sickle cell, yeah, sickle cell, yes.
yeah, yeah. So I wish that I can come back to my high school and learn this. This is very important, don't you think?
It is, yes.
Oh my
Um,
God.
yeah, I I can see what you're saying with the left and right. Normally when it comes to that type of activity, it is equal. But I'd have to look into ADHD because I think it's just it's it's not that well known, like you said. It's still kind of being studied for the
Mhm.
causes, the developmental issues. So it could be mitotic related, but I'm not sure it would be.
Mhm, mhm, mhm. Yeah, this is a subject, of research. Oh my goodness. I'm hoping this, okay, more kids coming to start taking this class. And then, okay, we are talking about a future researcher. All right. The world just so imperfect. Even in the US, okay, 3, 4 years ago, okay, they say there were like 6 million kids, okay, affected by ADHD. 6 million,
Hmm.
okay, that FDA, okay, all right. So can we imagine in the total in the global world how many of them? And there's no cure. Okay. So can we intervene during this growth when it's cell and cell? Okay, let's say they attach. Is there any way that we can, okay, in some way you like a CRISPR, okay, kind of, intervene so this bad thing will not happen and if everything in equilibrium? Oh, that will become a moral
So
issue, right?
yes, you remember. I remember you told, or was it you that told me that there was a man arrested because he CRISPR'd on his daughter? He gene edited his daughter. Yeah. So what he had to do was during development, like before the fetus formed, he actually had
Yes,
to go
yes,
in and do mitotic CRISPR stuff with mitosis. So yes, that's what he did.
you're
And
right.
as of right now, that's not ethical. That's not something that anybody agrees with. That's
Yeah,
why
yeah,
that
yeah,
he has so many. That's why he got so much trouble.
yeah,
But he actually went in and changed his daughter's material around during this mitosis stuff.
yeah,
The
yeah,
CRISPR, yes.
yeah. So the first case is in China. Okay, they try to protect the a daughter. A parent asked the scientist to protect her daughter, from AID disease.
Hmm.
Okay, so he manipulated. When he presented, he ended up to go to jail.
Hmm.
So the whole world, okay, in shock. How can we make a superhuman? I mean, this is a moral issue. Is there a moral issue or human issue? I
It's
mean,
it
yes.
it's so first of all, it was easy. So the one man did it by himself. So it's not impossible. It is sort of easy. And if you can do this, what are the limits, right? What can you put into something and what can't you put into someone?
Hmm. Yeah, 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 yeah. Wow. So I think next time, I think this is a very important stuff. It's not just for the doctor. It's for the general population. Okay, so this is what I'm trying to do. We hope, okay, in the near future, 2 or 3 months, we'll have this general kind of yeah, lectures about this. Okay, if you don't mind, because people need to understand. Okay, if smoking become, like having food or dinner. And even worse, it go like a train. Boop, 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 boop,
Hmm.
boop. Okay, pack after pack. Can you imagine what happened with the whole population? I
Yeah.
mean,
So there are foods too that can cause cancer.
yeah,
Uh-huh.
yeah. And it costs very expensive, in terms of for the country, to the health, okay? The health cost.
But
Yeah,
that goes back to um there was a whole issue with the secondhand smoke in the US and the government. Our government was making a lot of money on tobacco, right? They were making money from tobacco
yeah.
license, growth taxing. So
Taxes.
they actually kind of put the halt on all that information going out to the public.
Yes.
Yeah, so it's not easy, but it's not impossible again.
Yeah. But the thing then, all right, for individual level, okay, they need to understand and take care of themselves. If
Hmm.
the government cannot do it, okay, if somebody smoke, you just go away,
Hmm.
okay, not try to get close. But the fact of the matter is this, okay, if the parents smoke, that is the issue. The kid has no choice.
That
That
is
you
true.
even kill all of them. But this is a public health issue. It is a public not health not it is not an issue anymore. It is a crisis. It is a crisis everywhere except in the US, right? Even in the US, we always heard how this you know the 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 company, okay, the cigarette company, all right, try to, okay, influence the Congress or the
Mhm.
lawmakers, okay, try to get rid of any law or regulation through the lobbyists. You see, they pay million dollars for the lobbyists. Can you imagine
Mhm.
how to make poison, how to poison people, okay, lawfully?
That's
It's very
exactly what
sad.
happened. Yes.
It's very sad.
Hmm.
Okay. Oh my goodness. Okay. I hope that can. All right. We will be able to make, some of the population aware about all these things. Okay. With the consequences.
Yes. Then
Usually the cigarette company is a very strong and have a lot of money and to pay lobbyists, to pay politicians, and to pay everything.
you
Okay.
also have the issue where, like, we come from a rural area from here. Like, we're in the rural area. A lot of
Mm.
individuals 20, 30 years ago didn't go to uh higher education. They stopped at high school or even middle school. So when all this information was coming out about smoke, they didn't believe it, right? They didn't accept it. They didn't believe it. So more educated individuals understand it better and they accept it. But individuals that don't have the background, they don't believe it. They don't, again, my grandma smoked for 20 years and she didn't die of cancer, right? So why am I going to die of cancer? It doesn't mean
Okay.
that you will, but there's a high chance that you will get it.
Yeah.
That's one issue that we have too, especially
Yeah.
around here where individuals don't go and get higher education.
Yeah. So it is very easy to collect that information. All right. Whoever that get affected with, you say that, say a certain type of cancer, and look whether or not there's a relationship connection with the education level.
No, you
Like you
could, yeah.
said, in the lower level, in the very rural area, we expect they will be higher, get all that cancer thing, compare with, educated one.
Mm-hmm.
Okay? So that will be a very good paper for you to publish.
You've had time. So, okay, let's look at mitosis. This is the cycle of the cell where it was going to go through active splitting. So the purpose of this is to try to get those chromosomes that we just created and doubled to their own cells. So we have four subphases during mitosis: prophase, metaphase, anaphase, and telophase. So we'll start with prophase. Now, prophase is the phase that's coming right out of interphase. So you should see some characteristics at the very beginning with interphase. You still see a nucleus. The chromatin is still kind of visible, but the chromosomes are starting to form. And then we have these things called spindles. Spindles are going to start moving away from each other. And as they do, they produce these long filaments called microtubules. Now, towards the end of prophase, the nucleus is completely gone now. So you know you're in a mitosis phase when you don't see a nucleus. And then you have the microtubules, they're formed from the spindles, and then chromosomes are just completely randomized. So these two are showing you what's happening during prophase. We see our nucleus, chromosomes in the middle, spindles right here, spindle right here. And then later on at the end of prophase, my spindles should be on the complete opposite sides of each other. And then my microtubules are grown. And then you see these sisters in the middle, they're completely randomized. Now, from
Mhm.
here to get to the next phase, metaphase, remember these kinetochores that we just talked about? The kinetochores? The
Mhm.
microtubules in the first phase are going to grow and they're going to connect to the kinetochores on the sisters. And then from there, they can push or pull depending on where they landed. But they want to make all the sisters align down the middle, perfectly in alignment down the middle. And once we're in alignment, that's metaphase. That's the easiest phase to recognize. And then also down here, you can see these chromosomes, the blue ones. They're aligned
Mhm.
perfectly and almost in a straight line down the middle of the cell.
Mhm.
Okay, from here, we go into anaphase. Now, anaphase, the whole purpose is to move the chromosomes away from each other. So
Mhm.
on this side, I have a spindle. On this side, I have a spindle. Think of the spindles as fishing reels, and then my microtubules as a line, and the hook is on the sister. So when these spindles start to reel in the microtubules and pull them, the two sisters are going to be ripped apart. So one sister needs to go to one side and the other sister needs to go to the other side. They should not go to the same side because if they do, that's going to mess up the DNA content because each cell should be identical. And if I put two chromatids in the same cell, then I'm not going to have identical cells at the end of mitosis. The last phase is called telophase. Now, telophase, the way I like to tell my students is it's reverse prophase. Whatever happened in the very first phase, you see the opposite happen in the second phase. So chromosomes condense back down to the liquid chromatin. The nucleus disappeared. You can see it over here. The nucleus is reforming. And then my spindles appeared with microtubules. In the telophase, the microtubules disappear and the spindles go away. So it's completely reverse prophase. Also, during telophase, that's when a process called cytokinesis starts. So right down the middle of the cell, like right about there are proteins that are in the plasma membrane. They're called actin. They're going to be pulled on, and then as they get pulled, they're going to act like a rubber band. Have you ever put a rubber band on too tight, it cuts off your circulation? Same thing in sense here. The protein, as it starts to get tight, is going to start cutting the plasma membrane. So you can see it right here. The plasma membrane is being cut in half. And eventually they get pulled so tight that it separates and it pinches.
Hmm.
Whoops, what's going on? There it goes. So just a little bit more on cytokinesis. So right here, this dotted line, that's where my actin is getting activated. So that dotted line is going to be pulled tight. And then as it starts to pull tight, it makes this area called a cleavage furrow. So cleavage means to cut or to sever. So that's what it's going to happen. That cleavage furrow is the area where the cell is going to be severed into two new cells. So we're animals, and with our animal cells, we don't have that cell wall. We looked at this a few weeks ago. So when we go through cytokinesis, we're done at the end of cytokinesis. So right here, these are two individual cells. They're not connected anymore. However, with cells that have cell walls, like plants and fungi, bacteria, they actually have to go through another type of another activity after cytokinesis. So the cytokinesis is occurring right here. So when this one cell went through mitosis, it made two new cells, but the cell wall still has both nuclei contained in the same cell, and it wants to be two separate cells. So to remedy this, a cell plate forms between the two, and then when the cell plate forms, it's going to harden, and when it hardens, it makes two new cell walls. So now these two individuals, after that cell plate hardens, are two separate individuals. Now, this is only going to be important for things like plants and fungi and stuff like that. Okay, so mitosis is the sole purpose behind cancer cells. Now, when your cells, when they're working correctly, they have these things called checkpoints, and the checkpoints act as a go to the next phase. So you can see there's a checkpoint in G1, there's a checkpoint in G2 here, and there's a checkpoint in the mitosis phase. So each one of those little boxes is a checkpoint. So the cell's actually going to stop very quickly during their cycles and check for certain things. They go down their checklist, and if they pass all their checks, it allows it to go on to the next phase. Now, if for some reason a check fails and it decides that it should not go on, the cell actually can stop. And the way it stops is called apoptosis. So apoptosis is a special type of process where the cell, it hits a self-destruct button. It's going to say, I don't need to go on anymore. The cell does not need to be used anymore. And then it hits the self-destruct button and it terminates itself. So it does this. Once it hits that button, it creates some acid, some enzymes, and it kind of dissolves it from the inside out, and then it pops. So when the popping actually happens, it's apoptosis. Now, your cells actually do this a lot. So it's a good thing for apoptosis.
Yeah, the cell can test itself whether or not it can go on or stop, huh?
Correct. Yes.
Oh my goodness. I mean, it's like they have a brain.
They
Okay.
do. Yes. They had their own little um what's like security system.
Yeah, 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 yeah. Now, is there any research or advanced research, okay, how to make a certain cell, let's say a bad cell, all right, we affect the bad cell, okay, and say, okay, you need to stop now, instead of it going by its own brain. So we affect the brain to think. If it's a bad cell, okay, you stop.
Mhm.
Okay, you do what is it? Apoptosis.
Apoptosis. Mhm.
Apoptosis itself. Is there any research going that way already?
Yes, there is. So, um, see this right here, the tumor suppressor genes.
Mm-hmm.
So tumor suppressor genes are a type of
Ah,
checkpoint protein. They when working correctly, they stop the cell. So the way I tell my students is it's like a brake on a car. If the cell needs to stop, it finds out it's a bad cell and it needs to stop. The tumor
ah,
suppressor gene will slam on the brakes and the cell stops. So BRCA1 while ago with the breast cancer gene, that's a tumor suppressor gene. When it works correctly, it stops the car. Same thing with this one down here, the P53. It's also a tumor suppressor gene. So when it's working correctly, it stops the cell. But
yeah.
if you look at the other image I have over here, you see this big chunk out of it? That means the protein's been mutated. It doesn't work anymore. So you can think of the brakes have been cut. So
Mm-hmm.
the protein still exists and it's trying to slam on the brakes. But when it's doing that, it doesn't stop. The cell is going to keep going, and that's how it becomes cancerous. Now,
Okay.
you just asked if we can fix it, right? There's
Mm-hmm.
evidence, and we've done this a couple times, where we can use gene therapy. So we take some virus components. So the way a virus works, it affects the cell with their DNA. So if we have a gene that's broken, like the BRCA1 gene or the P53 gene, we can take a working, like new breaks, right? A new gene, insert that into a cancer cell, and it'll be like putting new brakes on a car.
How can make this tumor suppressor more in one person?
Well, I mean,
With
everybody has their
protein.
own. So you're like, it's scary, but your cells do this all the time. You're like, when your body's working correctly, you're
Ah,
yeah. So
and
you have
automatically you will have it.
you've probably done this several times already yourself.
Oh,
With
okay.
skin cells, with intestinal cells, those highly mitotic cells, each one have their own breaks, and everybody should be working.
Oh, so it is very important. I mean, it just, for me, it is truly amazing. It, you know, it, our body basically take care of itself.
Hmm.
Okay. So the question is this: what make our body stop thinking? Okay, or doing what it's supposed to be? What kind of thing that make we mentioned about it, right? One of them is smoking.
Hmm.
Okay, the other one is alcohol.
Alcohol,
Not mentioning but drug. Okay, and all that kind of stuff. So basically, when people do drug, they kill themselves for sure.
that is
Okay.
exactly right. Yes.
Yes, yes, yeah. We have to write this in a popular, in basically in a popular writing or article. But why people, you know, do drugs and do even the smoking, and a very simple cigarette thing. They keep doing it, but they do not know that really try
Well,
to kill themselves.
yeah, it goes back to brain chemistry with some of them people, though. Like I said, with nicotine, it's an addictive chemical,
Mm-hmm.
and nicotine plays off of our dopamine receptors. So they're
It
putting that
kills.
chemical back in their body because they they can't live without it. I mean, they think they can't live without it just because they had it so much. They
Ah,
can live without it. It's just going to take time to adapt and feel better. But the fact that they keep wanting to put it in their body, that's addiction, and
yeah,
that's brain chemistry.
yeah, addiction, and
Mhm.
that causes more trouble the more you do it. Okay,
Exactly right. The more you
after
do it, the
years
more
or
your body wants it.
so, after the body cannot hold it anymore, then hey, you have all this cancer. Okay, and that's it. And it's very expensive. You go with chemo, okay, insurance will pay. Basically the healthy people will pay it.
So I said this a while ago, but the best type or the best way to survive cancer is early detection. So the smaller you find the cancer, the easier it is to remove. The longer it's in the body, the bigger it gets, the harder it is to move.
It is. It is. It is. Yeah. Yeah.
And
Yeah.
if you think something's cancerous, it's very easy to determine. It's called a biopsy. They'll take a few cells, just a little chunk of cells off of what you think is cancerous,
Yeah.
and they'll check those cells. Because if the entire thing's cancerous, every cell in the mass is going to be cancerous.
Right. 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 Right.
And then this is the last slide I have for today. So these are just some ways that if somebody does have cancer, to treat cancer. So again, if it's benign and small enough, the go-to is going to be to get it out of the body. So just to surgically remove it. And
Mm.
you need to get rid of every cell to get rid of the cancer. Even if one cell stays around, it'll grow right back.
Yeah. Yeah. Yeah.
If if that's not an option, say it's malignant and it's in the entire body, chemotherapy is kind of the go-to here in the United States. So chemotherapy uses medicine, and the medicine is going to target mitotically active cells. And
Mm.
it's not pick or choose, it's every mitotic cell in the body. So the
Oh
goal
my.
here is to kill off the good cells in your body with the bad cells in your body. So somebody that's
Mm.
on chemotherapy, they're killing the cancer cells, but they're also killing hair cells and skin cells and teeth and gum cells, bone cells. Those are
Everything.
all highly mitotic cells, and the medicine can't be choosy. It's got to kill everything that's mitotic.
Yeah.
And hopefully with that, it kills the entire cancer.
Yeah. Yeah. So that is a very common treatment right now.
It is. That's one of the go-tos. Radiation therapy is another one. With radiation therapy, it's very similar to sunlight. It's a high energy beam. This high
Mm.
energy beam is enough to destroy the nucleus in the cells. And if you destroy the nucleus, you destroy the cell. The
Mm.
problem with radiation is it's spot treatment, so you can't do the entire body. And the deeper the cancer is in the body, the harder it is for the radiation to pierce it. So
Right.
it's
Right. Yeah. Yeah. So there's still a lot of opportunity here. Oh my goodness. It's about.
this last one though is what I find amazing. If this is the one that I think if we can perfect, this might be a cure-all for most cancers. It's called immunotherapy. So the way your
Mm.
immune system works is your immune system adapts to bacteria cells and viral cells, and it learns from them and kills them themselves. So if you can learn from a cancer cell, so if you can like map a cancer cell and you find a specific protein that's just on a cancer cell, you can teach your body's immune cells to look for that protein. And if it finds that protein, it will kill it. So you can
Mm.
basically teach your body to kill the cancer cells for you. The issue
Wow.
there is you have to have the cancer cell have its own protein that's specific to the cancer cell, and you have to have a cell in your body or somebody. You can actually take cells from somebody else's body and use those cells to teach your body to kill the cancer cells for you.
Yeah. Yeah. So there are so many type of cancer. Therefore, there are so many opportunity because to find the protein, okay, a specific protein only good to what? To treat a specific cancer.
Yes, that's exactly right. But
Right.
the thing there is, we have over thousands of different antibodies in our body, and we if any of those match a cancer cell, we can use those to fight off a cancer cell.
Yeah. Yeah. So there will be a more lab work, and all kind of thing. It's just fascinating. Okay,
Hmm.
I think this is the way how we need to go, I guess. Okay, if we want to make this world a better place to stay. So this is not, you know, between you and I, Ken. It is for the future researcher to do this. Okay, because they have thousands and thousands of that. Okay, that has not been found.
Hmm.
Okay, so
Exactly
we're
right.
hoping. Okay, some of these kids, all right, even though we have three, okay, consistently come, hopefully, okay, they will come out with the three, okay, uh, three type of approach to get rid of diseases, okay, okay. Thank you so much. It's exactly 9 o'clock, Ken.
All right, I'll see everybody next week. Y'all have a good day.
Yeah, thank you.
Welcome.
Thank you,
Right.
sir.
Thank you also.
Okay,
Goodbye.
bye-bye.
Bye.
Okay,
Okay,
now,
he's gone.
okay, you see that. Alfonso, Indah, and Hani. Oke. Jennifer, you can open your cam. Kamu dengar nggak tadi, Alfonso? Apa
She's
yang kita
connecting
diskusikan
to the
tadi?
audio.
Dengar nggak, Alfonso? Indah dengar nggak apa artinya yang kita diskusikan tadi, Indah? Coba. Ya. Itu Alfonso. Mama kan lagi di belakang itu pasti dengar kan? Mama dengar apa tanya? Mama duduk di belakang itu, kamu lihat tuh. Ibu.
Enggak. Boleh ya? Sekali.
Oke. Coba tanya ibu dengar apa? Oke. Indah, kamu dengar nggak apa, Indah?
Tentang about ADHD and the cancer and the cigarette.
Hmm. Apalagi yang kamu pelajari?
Tentang apa itu mitosis?
Nah, in general saja, in general. Secara umum, ya, setiap orang kemungkinan akan dapat cancer.
Ya, kuningnya doang.
Ya. Nah, tapi ya ada kemungkinan juga bagaimana ya seorang katakan peneliti, oke, atau seorang yang bekerja di dalam bidang kedokteran itu menemukan obatnya, bukan obat, ya kan? Tapi menemukan apa tadi? Protein, ya, protein. Nah, protein ini bisa menyembuhkan kanker sebelum dia muncul. Oke. Oleh karena itu ya, oleh karena itu peran daripada seorang dokter ya atau mereka yang berkecimpung dalam bidang kedokteran ya, ini sangat sangat diperlukan di dunia ini. Oke, sangat diperlukan. Bukan berarti kalau misalnya mau jadi chef, ya, Claudie kan mau jadi chef, seperti Chef Arnold, ya, tidak berarti tidak penting. Itu semuanya penting, ya kan? Kalau Xiao Chen itu masih nggak tahu Xiao Chen mau jadi apa itu, oke, ya. Tapi kalau ini pilihan menjadi dokter itu luar biasa. Kalian harus dengar cerita,
Saya jadi dokter.
ya. Mau jadi apa, Xiao Chen?
Dokter.
Nah, kalau kamu mau dokter, kenapa kamu nggak ikut biologi ini yang jam 8? Barusan itu kamu dengarkan tadi, itu profesor dari Amerika yang ngajar. Kalau mau jadi dokter, ya, kenapa nggak ikut? Gitu. Oke, kamu juga kasih tahu mamanya itu bahasa Inggris juga kamu jarang ikut, ya. Jadi kembali ya, adik-adik ya, ya.